semester. In going through the structures of the eye, we've got various eye models where we're going to see these various structures. This is the basic one, so the most common one, and it, for the most part, just shows you structures of the eye itself without a lot of what are called accessory structures of the eye. When we look at the surface of it, right, now we have an anterior view, we can see this something over here that seems a little bit odd. This actually represents the lacrimal gland. So the lacrimal gland produces tears, secretions to wash the surface of the eye, that then go from over here, which is the lateral side. The lacrimal gland is always located on the lateral side. The tear secretions wash across the cornea of the eye and the surface of the eye, and then drain into the lacrimal apparatus. So there's a lacrimal sac and lacrimal ducts, etc., where this is going to drain in on the medial side, in by the nose. But that's what this represents, in as much as this model can show it. This is the lacrimal gland. Other surface structures that we do see some of the various what are called extrinsic eye muscles. So extrinsic on the outside, that indicates that there's going to be some intrinsic muscles to the eye as well. So these are all skeletal muscles that allow the eye to look in different directions in a voluntary control. When we look at it, you can see that this is the inferior surface of the eye, right? This is superior, this is medial, and this is lateral, because we said the lacrimal gland is located on the lateral aspect of the eye. So these ones here that are located kind of orthogonal 90 degrees to each other are all straight muscles of the extrinsic eye muscle group. So this one's actually called superior rectus, this one's called inferior rectus, lateral rectus, and medial rectus. There are two other muscles that produce kind of angular movements of the eye. This one right here, you're seeing the tendon. This is the superior aspect of the eye. This is the tendon for a muscle that attaches at an angle, but then goes back and makes a path on the medial aspect of the eye, which is called the superior oblique. There's also one that's called the inferior oblique that you see here that attaches on the lateral side and then curves and goes inferior to the eye. And we'll see that in one of the other eye models where it shows the eye in the socket. But collectively, those are known as the extrinsic eye muscles. In terms of other things we can see on the surface, all of this white layer that we see here on the eye is called the sclera. Notice that it is the outermost layer, and it's actually kind of continuous with this clear area on the anterior side, which is called the cornea. So this is the cornea, and this is the sclera. It's the outermost tunic or layer of the eye. It's often called the fibrous tunic, consisting of the sclera and the cornea. All right, so to see other structures, we're going to have to get into the inside of the eye. This is the posterior of the eye. This is where the optic nerve is going to be exiting. But we'll see that coming up. So we'll do our model dissection. Now we're seeing on the inside of the eye, and we can see, this is anterior here, we can see that here is our cornea. And in fact, if we take a look closer at this hole where my pointer is going through is the pupil. And then the brown area of this model is going to be the iris, right? So it could be brown, could be hazel, could be blue, could be green, etc. But that is going to be the iris. Now within the iris, there are muscles that will control the size of the pupil, because the pupil is just a hole. So there are radial muscles that can pull it open. There are circular muscles that can cinch down and close it, and that adjusts the amount of light that's going to pass through this pupil. So the light's first going to go through the cornea, then it's going to go through, so we can see that there's kind of a space in there. That's called the anterior cavity, right? It's towards the front, and it's a space. That's the anterior cavity between the cornea and the iris. So it's going to pass through that. Then it's going to pass through that pupil, and then from there, it's going to pass into the lens. So the lens is right here in this model. So there's the lens. And the lens is attached to a number of structures that we see here in black. There are what are called suspensory ligaments that attach to it, and those then attach to this, which is called the ciliary muscles of the ciliary body. So now we're starting to see interior muscles, intrinsic muscles to the eye, those that are within the iris that can change or control the pupil size, as well as the ciliary body or the ciliary muscles that attach to the suspensory ligaments for the lens. And the reason that we have those attachments and those muscles for the lens is that changes the shape of the lens, allowing a different focus of the light rays on the back of the eye. Depending on whether you're viewing something that's up close to you or far away, that lens is going to change shape. Now, both the cornea and the lens work to bring the light rays together so that you have a nice clear image back here on the projected part of the inner eye or the retina. But the cornea doesn't change shape actively, but the lens can. So this is where we do a lot of active focusing of the eye by changing the shape of the lens.
So when people have issues with like farsighted, nearsightedness,
Yeah.
is it a function of the lens having an issue, or is it a function of what attaches to the lens to change the way that light comes in?
Or is it an issue with the cornea? Because the cornea also, like we said, focuses light rays. So the answer is yes. Can be all of the above. One of the most significant things that happens through time is as we age, right, tissues that are normally flexible or supposed to be flexible become less flexible. And so that certainly happens with things like the lens, where it's supposed to be able, I mean, it's stiff as we do our dissection. Granted, it's preserved, you'll see and feel that it's stiff, but it's supposed to have some flexibility to it so it can change that shape. But through time, it becomes less flexible, and so even though you may have these ciliary muscles pulling on it, it's just not changing shape properly, not allowing you to focus. So there can be issues in terms of the focus, as well, there can be issues in terms of the eye shape. So you can have these structures doing their job and trying to focus all of our light rays towards the back of the retina, but if the eye is oblong, like oval shape, and it's further away, or it's oval shape in this direction and it's closer, then it's not going to be in focus again. So your focusing mechanisms are doing what they can, but they may be working against another structure which is going to be affecting that focal length. And oftentimes when I talk about it, I kind of relay it to like the projector that we see here in the lab, right? So that has to focus the image on the screen there. And if the screen is farther away or closer up, it's not going to be in focus. If the lens of the projector is also, not focusing problem or not focusing properly, then you're going to have a fuzzy image. So either of those things can affect how clearly that image is showing on the screen. So in that particular case, then the screen is like the retina, right? And then effectively the lens, right, of the projector is like our lens that we have in the eye. And so both of those things need to be working together to give us a clear image. Okay, so those are some of the structures that we see towards the anterior of the eye. As we move through and the light rays might move through the lens, it's going to go through this space, which is called the posterior cavity. Now, both the anterior cavity that we saw up here and the posterior cavity have fluids within them. In the anterior cavity, we have the aqueous humor. A humor is a fluid. And in the posterior cavity, we have the vitreous humor. And whereas the aqueous humor, as the name indicates, is a little bit more fluid, the vitreous humor, as we'll find in the dissection, is a bit more gelatinous. So it helps to transmit the light rays, but it also, to a certain extent, kind of fills this posterior cavity, this space, so we don't have just a bag with nothing inside of it in terms of our eye structure. So we said that this is the retina. Notice that when we look through the retina, we can see these blood vessels underneath. There's a middle layer right here that's called the choroid. So the innermost layer is going to be the retina. The middle layer is going to be the choroid, and that's where all the blood vessels are. And then the outer layer, as we mentioned, is going to be the sclera. Okay, so all of this represents the retina, and then this thin brown layer that we see right here is the middle layer called the choroid. It's also called the vascular tunic or vascular layer. All right, so towards the posterior of the eye, we can see a couple of unique structures. This right here is where the optic nerve is going to connect in. And so the optic nerve, where all of the different axons are exiting the eye to connect to the brain, as we saw when we were doing the cranial nerves, right? We saw an optic nerve, which is number 2. That's going to be connecting there and routing all of that sensory information into the brain, back to the occipital lobe. But where it connects to the back of the eye, there is what's called the optic disc, a flat area. Now, in this model represented by this open circle here, a flat area that has just all these bundles. It's almost like a cable exiting the eye. And so in that area, that optic disc is also called the blind spot because the retina is going to be an area and a layer where we have all of our different photoreceptors, our rods and cones that we'll be talking about in lecture. But in this area, there's so many of these axons that are exiting the eye at this point. There's no room for our photoreceptors. So in this area where we have the optic disc, it's also called the blind spot. Technically, your brain is actively filling in that area because it's not getting any input from the retina or from the eye itself. So it's filling it in actively all the time in order to, prevent you from having two black spots in your vision as you look around. The other thing that we see towards the posterior part of the eye here is a yellow area here, which is called the macula lutea. And in the center of it, we have what's called the fovea or the fovea centralis. Now, whereas the optic disc has no photoreceptors, the fovea in the center of this yellow area has the highest concentration of our cone receptors. Our cone receptors are used in highlight conditions, allows for color vision. They give us the really detailed vision. And in this area right here, we have a really high concentration of them. So if, for instance, you are looking at this, if you were to take it up and you're going to look at it closely to examine that detail, you will automatically orient your eye so that the light rays are coming in and hitting right on that fovea so that you can get the most detailed image available. So that's called the fovea, and it's in the center of a yellow area that's called the macula lutea. Okay, so that is what this model looks like in terms of structures of the eye. Now, we'll just review that in terms of other models. So for instance, this one, it's kind of a cool one as it tends to fall apart as I'm rotating it. What we see here is we can see some of the accessory structures of the eye, right? So these are the eyelids, which are called the palpebrae. The opening in between them is called the palpebral fissure. So that's what this gap is called right here. Here we can see that lacrimal gland, and we can see these little drainage points here that are going to allow the tears to go from the lacrimal gland and then drain onto the surface of the eye, like I mentioned. Remember, this is going to be lateral. Drain across the eye, and then drain into these two holes. There's two little black dots here and here, which are called puncta, that then drain into these lacrimal canals here, that then drain into the lacrimal sac. So this goes down into the nasal cavity. Remember when we were doing the skull and we saw that there was a small bone on the medial side of the ocular cavity? It had that little hole in it. So that was the lacrimal bone that has that lacrimal sac within it that drains into the nasal cavity. So here's that membrane that lines it called the lacrimal sac here. So it drains those tears into the nasal cavity. So that's why if you are watching some sort of sad movie, watching your Pride and Prejudice or whatever gets you sad, then it's not that when you're crying, all of a sudden your nose starts running. What's happening is, right, the secretion of tears from the lacrimal gland is really cranking up. It's washing across the eye, and it's draining into your nose. And so those are actually tears that you're sniffing back. And if those drains get overwhelmed by even more secretions, then of course it's going to be running down your nose, running down your face instead. Okay? So that's what this is showing us. This is a unique aspect of this model here. If we remove that, then almost everything else about this model is going to be the same. We see the extrinsic eye muscles. Here on this one, we can see the optic nerve sticking out the back. We can see here a representation of our choroid, our middle layer with the blood vessels. We can see our sclera on the outside. We can see our cornea here, the clear area that overlies. Removing it. The iris, and then the pupil, the hole in the center of the iris. If we remove this, this represents the vitreous humor. There's our lens. That's just posterior to the pupil. And then here, we can see those suspensory ligaments, like in white right there, and the ciliary body, represented in red. Now this, again, this innermost layer is going to be the retina. We can see blood vessels through it, but this innermost layer is going to be the retina. And then here is our optic nerve hooking in. And so here, a little bit clearer, we can see an optic disc where it actually connects. right? And if we look at this side, one of these sides should show us a fovea as well, a fovea centralis. Right now I'm not actually seeing it, but it should be just adjacent, oh, here it is, just adjacent to the optic nerve, okay? So we can see it over there. All right, so that's what it looks like on that model. So the only new things on that model represented the accessory structures that we saw associated with the lacrimal gland. The other interesting thing, as that eye is kind of looking back at me, is when we look at this model. So this model has superior rectus muscle jumping off, but this one shows it a little bit more anatomically in the socket, right? So we can see the nasal cavity here. There's that lacrimal sac that we were talking about, but it also shows us the pathways of those different extrinsic eye muscles, right? So superior rectus, lateral rectus, and here you can actually see there's the tendon for our superior oblique, but notice that as it comes across here, it goes through a little loop of connective tissue here on the medial side, and then this, right here, this pathway here, is where the belly of the muscle is. So it almost has a right angle, or even more than a right angle, here to attach to the surface of the eye. So that's why it's called the superior oblique, because it comes in an angle like that, but it uses this pulley, which is called a trochlea, to then loop back and connect to the rest of the muscle back here. So an interesting aspect about that, remember that when we were doing the cranial nerve, there was a cranial nerve called the trochlear nerve, right? The trochlear nerve sends impulses to the superior oblique muscle to tell the eye to move in a certain direction. So oculomotor nerve controls 4 of the other extrinsic eye muscles, but the trochlear one just controls this one. And that little loop that we said, like a pulley that helps to work like a pulley system, that loop is called the trochlea. So the trochlear nerve is named for that trochlea where the superior oblique cuts through. Okay? Okay. So other things we can see, there's that big optic nerve. And in fact, in this case, you can see it coming to the optic chiasma, the X, where the axons crisscross in order to go back to the brain. And then the rest of the internal structures of the eye basically look like the same model that we just went through, because it's made by the same company. It's called 3B. So it looks the same in terms of the internal aspect of the eye, but it is pretty interesting to be able to see the full pathway of those different extrinsic eye muscles. Okay? Okay, then finally with the eye, I'm gonna have to zoom out for this one. Oh, that's really big. We see this one, which is a parasagittal section of the eye. So I'm going to move this out a little bit. Hold on, don't go anywhere. There we go. So this just gives us a parasagittal cut of the eye. So it gives us a different view of the structures, and it allows you to kind of see the way that light rays would be passing through the different structures of the eye. So there's the cornea. If we continue to pass that around, this is going to be the sclera, the white of the eye, right? So there's the cornea. There's the anterior cavity that the light would pass through next. Here's the pupil, the hole within the iris. There's our lens, and we can see our suspensory ligaments attaching to it, represented in white there, and then this is the rest of our ciliary body that will act, and this is also the ciliary body here in cross-section, right? Because we're cutting through that whole loop of muscle that surrounds the lens. And then we get into the posterior cavity. In terms of layers, we can see the innermost one here is going to be the retina. Remember, the middle one is the cornea, and then, I'm sorry, not the cornea, the choroid, choroid, and then the outer one still is going to be that sclera. We can see the optic nerve hooking in the back. You can also see it's a pathway for blood vessels right through the inner part of it that then spread into that choroid. So there's the optic nerve, which means that this area here is going to be the optic disc or the blind spot. And then this represents our fovea centralis that we see right there, that specialized area of the retina, which has what's called high visual acuity because of the high concentration of cone photoreceptors. Okay? So nothing different on this, it's just a different view of it. So you want to make sure that you review the structures of the eye as well as some of those accessory structures in the different models that we have so that you can recognize them regardless of where you're being asked. Okay? Alrighty, so those are the structures of the eye.